Your fat cells are supposed to die. That is how healthy tissue works. Old cells clear out, new ones take over, and your body renews itself. But research published in peer-reviewed journals has confirmed that in visceral fat, the fat packed around your organs, a growing population of cells has stopped dying. They have stopped functioning, and instead of clearing out, they are broadcasting a chemical signal that is keeping your fat tissue permanently inflamed and locked. They are, in the most literal biological sense, zombie cells. Scientists call them senescent cells. They are not alive in the way a healthy fat cell is alive. They cannot divide, cannot respond to your metabolic signals, cannot be cleared by dieting or exercise. But they are not gone either. They are sitting there, releasing a constant stream of inflammatory chemicals into your surrounding tissue. And here is the part that makes this so relevant to anyone who has struggled with stubborn visceral fat, especially if you are over 40. These zombie cells spread. When a senescent cell releases its inflammatory signals, a mixture of chemicals that researchers call the senescence-associated secretory phenotype, or SASP, it does not just affect itself. Those signals reach neighboring healthy fat cells, and research published in peer-reviewed journals has shown that those signals can push healthy cells into the same dysfunctional state. One zombie cell becomes two. Two become four. And every cell that enters this state does the same thing. It locks the surrounding tissue in a chronic inflammatory environment that makes fat mobilization significantly harder. This is not a new fringe theory. Research published in journals including Cell Metabolism, Aging Cell, and Geroscience has been building this picture for years. What is relatively new is the understanding of how aggressively this process accelerates after the age of 40 and what you can actually do about it. In this video, I am going to walk you through exactly what these zombie cells are, how they accumulate in your visceral fat, how they create a self-reinforcing inflammatory loop that makes visceral fat resistant to reduction, and what the current research says about interventions that may help clear them. If you have been frustrated with fat loss that stalls despite doing everything right, stay with me. Because understanding this mechanism changes the question you should be asking, from how do I eat less to what is making my fat cells stop responding. Every cell in your body has a life cycle. It divides, it functions, and when it reaches the end of that life cycle, it is supposed to trigger a process called programmed cell death, apoptosis, and clear itself out. New cells take its place. The tissue stays healthy. Senescence is what happens when that process breaks down. A fat cell becomes senescent when it accumulates enough damage from oxidative stress, from DNA damage, from exposure to high levels of insulin or glucose over many years, that it triggers a cellular arrest. Think of it like a factory machine that has overheated and locked up. It cannot produce anything useful. But unlike a machine you can simply switch off, a senescent cell does not shut down cleanly. It stays powered on, and it broadcasts. The SASP, the senescence-associated secretory phenotype, is that broadcast. It is a continuous release of inflammatory cytokines, including IL-6 and TNF-alpha, along with matrix-degrading enzymes called matrix metalloproteinases, or MMPs. These are the same molecules that drive chronic low-grade inflammation throughout your body. Research published in the Journal of Clinical Investigation documented that visceral adipose tissue, the fat depot surrounding your internal organs, is particularly susceptible to senescent cell accumulation compared to subcutaneous fat, the fat sitting just under your skin. In studies measuring senescence markers in human adipose tissue across BMI ranges, researchers found a consistent pattern. The higher the visceral fat burden, the greater the concentration of senescent cells. This creates a situation where visceral fat does not just passively accumulate. It becomes an active inflammatory organ, driven in part by the growing population of zombie cells inside it. And critically, for anyone over 40, this process is not random. Research published in Aging Cell has shown that the normal immune clearance of senescent cells, which your immune system is supposed to handle, becomes progressively less efficient with age. The cells accumulate faster than your body can remove them. This is why visceral fat often becomes noticeably more stubborn in your 40s and 50s, not just because of hormonal changes, but because the cellular landscape inside that fat tissue has fundamentally changed. The spreading mechanism is what makes cellular senescence so relevant to visceral fat specifically, and it is what distinguishes this from ordinary inflammation. When a senescent cell releases SASP factors into the surrounding tissue, those molecules do not just cause local irritation, they act as paracrine signals, meaning they travel to neighboring cells and alter their behavior. Research published in Aging Cell documented what happens next in adipose tissue. The SASP factors from one senescent cell can impair the differentiation capacity of neighboring adipose progenitor cells, the cells whose job it is to replace old fat cells with new, functional ones. When that replacement process is blocked, the tissue cannot renew itself. Old, dysfunctional cells accumulate, and some of those cells, exposed to the chronic SASP environment, become senescent themselves. The cycle continues. Think of it this way. Imagine your visceral fat tissue as a neighborhood. A healthy neighborhood has houses being repaired, old structures being replaced, and everything functioning as it should. Now, imagine one house catches fire, not a destructive fire, but a slow, smoldering one that never goes out. The smoke drifts to the houses next door. Over time, those houses start smoldering too. The neighborhood does not burn down quickly, it just slowly stops working. Repairs stop, new buildings do not go up, everything decays in place. That slow smoldering is your SASP environment, and the decay is what we see metabolically as resistant visceral fat. The key mechanism here involves two specific inflammatory cytokines, IL-6 and TNF-alpha, which SASP cells release continuously. These cytokines do several things that directly impair visceral fat metabolism. They suppress adiponectin, the hormone your fat tissue is supposed to release to signal fat mobilization. They promote insulin resistance at the level of the fat cell itself, making the cell less responsive to lipolysis signals. And they recruit immune cells, particularly macrophages, into the visceral fat tissue, which then contribute their own inflammatory output, amplifying the original SASP signal. Research published in Biological Research in 2024 described this as a pro-inflammatory state and exacerbated oxidative stress cycle, specifically noting that the expansion of visceral adipose tissue is characterized by an increase in senescent cells and impaired differentiation of pre-adipocytes, creating conditions where fat reduction becomes progressively more resistant to standard interventions. If you are over 40 and have experienced a plateau that makes no sense given your diet and exercise effort, this mechanism is worth understanding. It does not mean your fat loss is impossible. It means the obstacle may not be caloric. It may be cellular. This is the section that actually matters, because understanding the problem only helps if there are evidence-supported strategies to address it. The most extensively studied intervention in this field is a class of compounds called Senolytics, agents that selectively target senescent cells and prompt them to undergo the apoptosis they were supposed to undergo in the first place. Think of them as a targeted clearance crew for zombie cells. The most researched senolytic combination consists of two compounds, dasatinib, a pharmaceutical agent, and quercetin, a plant-based flavonoid found in foods including onions, apples, and capers. Research published in eBiomedicine documented the first human pilot study of this combination and found measurable reductions in senescent cell burden in human tissue. Subsequent research published in Aging Cell in 2023 specifically found that this combination attenuated adipose tissue inflammation and improved metabolic function in aged subjects. A second natural compound with growing research support is ficitin, a flavonoid found in strawberries. Research published in Mechanisms of Aging and Development in 2024 reviewed the clinical evidence for ficitin as a senotherapeutic agent, noting that in a recent human study, ficitin reduced the numbers of senescent peripheral blood cells and reduced circulating SASP factors. Multiple ongoing clinical trials are investigating its application. However, and this is the honest caveat your research-based approach requires, the majority of strong mechanistic data for Senolytics in visceral fat specifically comes from animal models. Human clinical trials are underway but limited in scale. The Endocrine Reviews Journal noted in a 2024 review that while preclinical data appears promising, clinical evidence remains preliminary. Quercetin and ficitin are generally considered safe at dietary and supplemental doses, but pharmaceutical senolytics like dasatinib carry clinical risks and are not appropriate for self-administration. Beyond senolytics, there is a second intervention pathway with growing evidence that is accessible to everyone: intermittent fasting. Research published in Geroscience in 2024 examined the effect of intermittent fasting specifically on senescent cell populations in aged adipose tissue. The study found that intermittent fasting reduced senescent phenotypes in adipose stem and progenitor cells and restored their adipogenic capacity, meaning the fat tissue's ability to regenerate healthy cells improved. Separately, a human exploratory study published in Human Nutrition and Metabolism in 2023 documented that 30 days of prolonged intermittent fasting showed a tendency to reduce expression of the senescence markers P16 and P21 in the blood of participants. This connects directly to the fasting mechanism you may already know about, autophagy. When fasting activates autophagy through AMPK and ULK1 pathways, it does not only clear damaged organelles, it appears to selectively target and clear senescent cells, a process referred to in the research literature as geroautophagy. Your fasting protocol is not just a fat-burning tool. It may also be a zombie cell clearance tool.
Based on the current research, there are 3 evidence-supported actions that align with what we know about visceral fat senescence. The first is consistent intermittent fasting. The Geroscience data and the Autophagy research both point toward extended fasting windows, particularly 16 to 24 hours, as a meaningful way to activate the cellular clearance pathways that address senescent cells. This is not about extreme restriction. It is about giving your body the fasting window long enough to activate geroautophagy. The second is quercetin through food. Onions, capers, apples, and berries are the highest dietary sources. The research on quercetin as a senolytic used supplemental doses, but consistent dietary intake of flavonoid-rich foods supports the broader anti-senescent environment in your tissue. Combined with ficitin from strawberries, you are building a dietary pattern that the research associates with reduced senescent cell burden. The third is managing the inflammatory inputs that accelerate senescence in the first place. Chronic elevated insulin, oxidative stress from ultra-processed food, and sleep deprivation all accelerate the rate at which fat cells reach senescence. Reducing these inputs does not clear existing zombie cells, but it slows the pipeline that creates new ones.
The research on cellular senescence in visceral fat is telling us something important. Stubborn visceral fat is not always a calorie problem. Sometimes it is a cellular environment problem, a tissue that has been taken over by dysfunctional cells broadcasting inflammatory signals that prevent normal fat mobilization from occurring. You are not fighting yourself. Your fat cells are fighting each other, and the zombie cells are winning by creating a neighborhood where healthy renewal cannot happen. Understanding this mechanism does not make fat loss effortless, but it changes where you direct your effort. Fasting, flavonoid-rich foods, and reducing the metabolic inputs that drive cellular aging are not just general health advice. They are targeted strategies supported by the biology of senescence. If this gave you a new way to think about your visceral fat, not as a calorie storage problem, but as a cellular biology problem, type yes in the comments. I want to know how many of you have been dealing with resistance that no amount of dieting explained. If you found this useful, share it with someone who is frustrated with fat loss that should be working but is not. They may be dealing with this exact mechanism. Subscribe for more evidence-based breakdowns, and I will see you in the next one.